Hey there, fellow primates. I hope this video finds you well. I hope you have been better than I have because I got the COVID. It's something that happened 16 days ago. I attended a dinner with friends and colleagues in a pub in London, and of course it was a big group of people. We hadn't seen each other for a long time. We talked, we screamed, we drank, we ate, and then a bunch of us seated at the same table got COVID, which was unfortunate timing because Easter weekend, which was the weekend after that dinner, I was supposed to have met up for the entire weekend with my friends Pascal and Jose and Pascal's sons Mark and Trevor, and I did on the Friday hang out with them, but then I started to get a sore throat, and on the Saturday I tested positive, so I couldn't see them anymore. Hadn't seen these people in more than 2 years, so that was shitty timing. But I was sort of lucky. For me, the worst symptoms were sore throat, was for 3 or 4 days, sort of slightly tight feeling in the chest, a bit of a runny nose, a few days of brain fog, which made it difficult to concentrate. You just sit there sort of staring into the middle distance, but luckily that was about it. Oh yes, and tiredness. Oh my God, I was so tired. And now it's 16 days after I was infected, and my lateral flow test today for the very first time was almost negative, a barely visible positive line. My ability to stay awake has been compromised. I have to go to bed still about an hour earlier than I used to before COVID because I'm just that damn exhausted. Could have been worse. My friend Jackie, she was knocked out for like 5 days or longer and was bedridden. So it really attacked her horribly. It's a shitty disease. But anyway, I'm back in the saddle, so to speak. Did a bit of reading as I usually do, but nothing stood out in the past week that I wanted to dedicate an entire discussion to. So I'm just going to highlight 3 stories that fall somewhere at different points along the spectrum of storytelling in science. So first paper I want to draw attention to was published in Molecular Biology and Evolution, an important journal, and it's titled Evolution of Gene Expression Across Species and Specialized Zooids and Siphonophora. Siphonophores, what a word, siphonophores are a group of cnidarians. They belong to the hydrozoans, the same group that the laboratory animal Hydra belongs to. And they are some of the most morphologically complex cnidarians. They have, for instance, Portuguese man-of-war, maybe a siphonophoran that you're familiar with. And their individual siphonophorans have bodies with different components that are different zooids. Some correspond to medusae, jellyfish-like body forms, some to polyps, and so some of the most complex siphonophores have a combination of these medusae-form and polypoid zooids that are functionally differentiated to provide different functions for the colony. Now, these are the opposite of model organisms. Many, I think all of them, are planktonic, very fragile creatures, difficult to collect, difficult to work with, and so very few people work with siphonophores. Even fewer have the cojones to work molecularly on them. One of these brave people is Casey Dunn and his team, and so they published this paper. Now, I was very excited when I saw this in my table of content alerts because not many people work on the molecular biology and evolution of siphonophores. Plus, these days, with little money, you can generate enormous amounts of omics data, which they did, transcriptomic data, and you can tell with the sophisticated techniques that we have all kinds of interesting stories. So I thought this is going to be interesting. And for instance, they in the abstract say that they found, for instance, that if you look at different zooids in different siphonophore species, gene expression patterns seem to be consistent between them. And they conclude that our findings show that patterns of gene expression have the potential to define zooids and colonial organisms. Cool. Tell me more. And they did. And then I got a little bit disappointed because the entire discussion and the entire presentation of results is quantitative, very quantitative. To give you an example of this, they say between the white mature gastrozoid and the yellow mature gastrozoid in B. elongata, few significantly differentially expressed genes were identified. 8 genes were up in white mature gastrozoids relative to 36 genes up in yellow gastrozoids. Among genes that were significantly differentially expressed in either white or yellow gastrozoids relative to all other tissues, 276 genes were unique to yellow gastrozoids and not found in white gastrozoids, and 886 genes were found in white gastrozoids and not found in yellow gastrozoids. That's interesting, perhaps, although as a non-siphonophore expert, and yes, they do present a schematic drawing of a siphonophore to give the reader a little bit of concrete, a picture to focus on. This is pretty well quantitative, difficult to remember. It doesn't get more concrete in this study than Go terms, and Go terms are gene ontology terms. You can look at genes expressed in the tissue, and the function of the genes generally has been studied only in very few model organisms like mice or Drosophila or C. elegans, the nematodes. And based upon functional annotations of these types of genes in these model organisms, you can extrapolate to poorly studied creatures like siphonophores, and then hope you can unwaveringly say something about function. What do they conclude? And this is as concrete as it gets. For instance, they say for female gonodendra, which is a type of zooid, there was a Go term enrichment for a number of biological processes, including mitotic cell cycle process, DNA replication in one species, as well as a number of signaling pathways and developmental processes in another species. This is not really memorable. There's not real anything that you can get narrative purchase on here. And that is a problem if you want to convey the results of your study to a reader. Take, for instance, this tree as one illustration of the type of diagrams they present. This is a gene tree of Wind genes, and you can see plotted on the branches positive numbers and negative numbers. And a positive number would be an increase in gene expression along that branch, a negative number a decrease. So if you look at the top clade, Wind2 genes, you can see all the species represented, and along some branches, Wind2 genes have been increased in expression and in others decreased. Well, you know, they looked at hundreds and hundreds of different genes, so this is a tiny little quantitative speck in an enormous mass of quantitative data. This is difficult to wrap your head around. It's difficult to get a take-home message out of it. It's difficult to find this engaging. It's not their fault. I think this is an almost, what you call it, there's a phrase for this, occupational hazard. That's the phrase. That you, as a scientist, collect enormous amounts of data, which these days can easily be done. You can collect in a few days so much data that you wouldn't be able to finish analyzing it for the rest of your career. And then just look at it in different ways, have bar diagrams and pie charts and histograms and whatnot, and just present the data and say, quantitatively, this is what gene expression tells us, for instance. But comparative gene expression studies presented purely quantitatively, doesn't really tell a story. And hence, for the human reader of such work, I have to say I didn't find it very interesting. And maybe, undoubtedly this is partly due with the type of data, but also partly due with perhaps the authors not really trying to tell a qualitative story that readers can have narrative purchase on. And I thought that was, a bit sad. It's an important paper. They develop a new metric for doing comparative gene expressions between studies between species. They develop a new method of looking at changes in gene expression across phylogenetic trees. Important, can be applied to other types of systems, but it was not really a story. It was a bit boring. What's different for the second paper that I read by Mark Pekel and his colleagues, published in Nature Communications, entitled "A General Statistical Model Shows that Macroevolutionary Patterns and Processes Are Consistent with Darwinian Gradualism." So macroevolution refers to evolution above the species level, the kind of things you infer when you compare species and higher taxonomic units. And this contrasts with microevolution, which are the processes and patterns of evolution you find within species, the level of evolving populations. And of course, certain evolutionary theorists, for instance, famously the late Stephen Jay Gould, wanted to develop a macroevolutionary theory that they hoped would be distinct, causally in some way disconnected from evolution on the population level, microevolution, because then, if you look at the fossil record, you can develop a body of theory that applies to species and higher level taxa without having to feel inferior because you cannot do population biological studies on your organisms. So the question has been, for instance, if you look at the evolution of animal body plans, as revealed in Cambrian fossil records, famously the Burgess Shale. You see the sudden emergence of these really weird complex body plans. Does this mean that some weird macroevolutionary processes that are very distinct from the gradual incremental evolution on species levels has created suddenly in a big bang these body plans? Or is this just due to preservational biases? Is macroevolution simply microevolution writ large or not? And this is the topic that Mark Pagon and his colleagues delved into. And they looked at 2,859 species of mammals, which they placed in a phylogeny, which you can see here. And they asked, how did body size evolution happen? Did it happen in, for instance, certain big bangs, very abrupt evolution that created suddenly from small ancestors big descendants? Or could evolution be captured by a normal Brownian motion, a random walk through available variation in populations that eventually produced all these different body sizes from mice to elephants? And so they compared different models that had been published in literature before and developed their own. And what they found was interesting. So if you look at the circle diagram, you can see different colors. You can see blue and red. And you can see in the center of the diagram two symbols, V and B. I mean, this is a beta. And I don't know, is it a me or a ni, the V? I don't know if it's a Greek or Latin alphabet, so Greek to me. So let's just call it a V and a B. The V designates the availability of variation available to a clade to evolve into. And the B is the step size, as it were, of directional evolution. So if the V is larger than 1, then there will have been at a certain node in the tree an increase in the amount of variation available for taxa lineages to evolve in. If the V is smaller than 1, that means that from an ancestral node there's suddenly been a contraction of the available variation that lineages can evolve in. And with the beta, if it's higher than 1, then there will have been an increase in the phenotype, in this case an increase in body size, and if it's smaller than 0, then there has been a decrease in body size. And you can see there's been an almost chaotic pattern of evolutionary changes of body size in the mammalian tree, in both increases and decreases of the variation available for lineages to evolve, and big steps in body size evolution, increases and decreases. And specifically they say there have been 417 such directional steps in body size on this tree, and 190 changes in the basically available variation, increases or decreases. Interesting. So they conclude that increases of evolvability, which is the increase of variation available to evolve into, and directional changes are decoupled. So that's an interesting finding. They're not coupled to each other. Another finding is that the branch lengths of this tree, which are calibrated to the fossil record, so they are basically estimates of absolute evolutionary time. The branch lengths do not associate at all, correlate with the amount of body size evolution that happened along them, which is a very interesting finding because people use models often to, for instance, estimate ancestral states in which they use these branch lengths as informative about whether or not evolutionary change has happened. And this particular study that shows that for body size evolution in mammals, branch lengths are not informative about whether or not there has been directional evolution in body size. And that's very interesting. And the third interesting thing that they conclude and reflect in their title, because that's the most general finding, as it concerns body size evolution in mammals, you don't need to posit any specific, distinct, macroevolutionary, weird type of evolutionary processes to explain even sudden bursts of body size evolution. They show that the step size of body size evolution, the variance needed to evolve body sizes, falls within that are made available by standard mutational variation. So yes, as it concerns body size evolution in mammals, Macroevolution above the species level is simply microevolution below the species level writ large, which is an interesting finding. Of course, this is one type of trait in one type of organism, so how generally microevolution and macroevolution can click together and you can assess one in terms of the other remains an open question, although really I don't think that there is any evidence for macroevolutionary patterns. I think that pretty much all macroevolution is microevolution writ large. But interesting findings anyway, an interesting story produced by them developing a new model that captures these different aspects of body size evolution. Last paper that I want to spend a few minutes on was published in the journal Developmental Biology by Eddie de Robertis and Nadia Tejeda Munoz. And it is a review article in a special issue, I think, that goes into the topic of dorsal ventral patterning in animals and what it says about the homology of body plans. And this is an interesting paper. Geoffroy Saint-Hilaire is mentioned 2 weeks ago. My last video was about whether or not there had been dorsal ventral axis inversion of arthropods relative to vertebrates, and whether Geoffroy Saint-Hilaire already in the 1800s came up with this idea. And these authors basically say yes, that he thought, Geoffroy that is, that arthropods and vertebrates are 180 degrees inverted with respect to each other. Of course, when you see my last video, that's an incorrect, at least historically incorrect conclusion. But they, and this is an interesting review paper, summarize all the evidence that shows that the molecular machinery that patterns the dorsal ventral axis in arthropods, Drosophila, and vertebrates, specifically Xenopus, which is the model system that Eddie de Robertis and his colleagues have done a lot of groundbreaking work on since the 1980s, are essentially composed of homologous components and are inverted 180 degrees, are homologous and oriented with 180 degrees difference with respect to each other. So that's interesting. But then, they propose a little scenario, which you can see here in this cartoon, that Urbilateria, which is the last common ancestor of all bilaterians, this is a word actually that Eddie de Robertis and the Sazai, I forget the first name of his co-author, was it 1996 came up with, or 1994? And with a quite similar sort of cartoon like this. And they say, you can look at Urbilateria. Urbilateria had an amphistomic blastopore, which is the yellow bit, where you have an opening in the adult, they say, that runs from mouth to anus. But that's a weird kind of creature. Amphistomy is actually, and this is a topic I discuss in my book at quite a lot of length, the type of blastopore that you can find in an embryo, but then in the adult, the blastopore closes in the middle, leaving a mouth in the front and an anus in the back. But they proposed that this creature had this one single large opening. And then in protostomes, it closed from the back and it retained the mouth opening, and in deuterostomes it retained the anus in the back and a new mouth broke through. And it's like, well, they say that the ancestor had this amphistomic body plan, which is interesting because amphistomy, if it even exists, is extremely rare, especially when amphistomy is defined with respect to the opening of the blastopore, as I discuss in a chapter in my book. If you redefine it as blastoporal tissue that surrounds the blastopore, giving rise to tissues that you find in the front end and the back end, then you can maybe redefine amphistomy as something that happens in a lot of creatures. But creatures that actually close the blastopore in the middle and leave a mouth and an anus open at either end are very hard to find in literature. Nevertheless, these authors believe that Urbilateria had that, and they do that because they have a scenario they sketch literally sort of on the back of an envelope based upon their own evolutionary intuitions. They didn't do character mapping. They concluded Urbilateria had a trochophore type larva. They didn't present real evidence for it. They just believed that evolution went this way. So this is a nice paper. Interesting summary of the molecular biology of dorsal ventral patterning in 2022 that uses the exact same storytelling tools based upon central convictions and intuitions that has been sort of the method of doing phylogenetics for more than a century and a half before cladistics and rigorous models of substitution models sort of revitalized, revolutionized is perhaps a better word, phylogenetics. Although in my book I argue that even the use of substitution models, that's a narrative way of telling evolutionary stories, just as it is to sit down with an envelope and a pen. But this is a story, it's storytelling, the old-fashioned way. One thing that stood out in the paper, and that is the kind of thing that makes reading papers sometimes so exciting, because it is a puzzling statement and you want to know more. They write, for instance, when the work by Eddie de Robertis and colleagues was published in the 80s and 90s, that flies and frogs have these homologous conserved dorsoventral patterning molecules in an inverted position. They said that in the 1980s, no one could have predicted that the development of species as different as fruit flies and the frog Xenopus would have a common developmental blueprint. And then on another page, they say, just there we go. So nobody in the 1980s would have expected that there would be a conserved developmental toolkit between arthropods and vertebrates. And similarly, nobody could have predicted the amazing degree to which the animal gene toolkit had been conserved in development. So in the 1980s, they claim nobody thought that such divergent body plans as arthropods have and vertebrates have could be based on developmental and genetic similarities. I go, really? Because in the 1980s, I know, although they don't cite him, this echoes statements that the great evolutionary biologist Ernst Mayr had made in a book in the 1960s. He says, it would be futile to look for homologous genes in distantly related species because you won't find them. But is that really the case? Was that really the case in the 1980s? It was in the late 1980s that the first molecular trees were published based upon 18S ribosomal RNA, and homologous gene, not just in animals, but within a much broader range of living organisms. Were biologists in the 80s really thinking that things that you could find in different bilaterians, such as coeloms, in very diverse phyla, central nervous systems, sensory organs, segmentation, that they were underpinned by non-homologous genes, because many of them thought that segmentation, for instance, in vertebrates and in arthropods, went back to homologous precursors in a bilaterian ancestor, like a segmented bilaterian ancestor. And if that was the case, wouldn't you expect at least that some of the molecular factors and the genes and the developmental processes involved in these were homologous too? So this is the kind of statement that really invites you to dig deeper into the historical literature, because really, were the threats of phylogenetic continuity thought to be absent in the 1980s, quite late, when we knew that DNA was the universal molecule of heredity? And I'm sure that we already had some inkling that the genetic ties underpinning at least animal diversity went much deeper than just closely related species. So I don't know, I would have to go into the literature. But that's one of these statements that you read in the paper and that the authors use to set up their argument, and they say, oh, that was a major surprise. Was that really so? Anyway, I've been waffling for 24 minutes. This is long enough. When I don't find interesting articles to discuss in depth, I don't know if I should do random paper discussions like this. I set up this channel mostly to scratch the itch that I feel to do Journal Club, but now that I'm going back to the museum a bit more, it is also great to actually do Journal Club with living colleagues. And B, this channel is also, this is not a secret, a sort of vehicle that I intended to help promote my book when it comes out later this summer. So I think in July I will do a series of short, maybe 10-minute videos of each of the chapters of the book, a central idea from each of them, just to get people's appetite whetted for the book, so they will actually buy, more importantly, read the book. But yeah, so this week a bit of waffling about 3 papers that stood out from the things that I've read. Don't know what I'm going to do next week. If you found this interesting, give it a thumbs up underneath. Sign up to the channel, sign up for alerts if you want alerts in your life. I hope that you are going to have a nice bank holiday weekend. If you are in the UK, you will have a bank holiday weekend. If you're not in the UK, you may have a short, normal 2-day weekend. In any case, I hope you're all okay. I hope you continue to evade COVID. And I'll see you next time. Bye-bye.